We'll be live in a second. All right, and we are live. Hello, everyone. Thank you for tuning in. This is the first live talk we're organizing here at Cascades Analytics on LinkedIn. This is very exciting. We have a fantastic guest today, a trailblazer. And I just wanted to give a few information on Cascades Analytics. It's a consulting bureau of neuropharmacologist experts based in Paris and New York. And so we provide insight on, especially psychedelics, neuropharmacology. And so what's better than, be able to talk to experts like Dr. Urosh Laban to understand the structure and function and, potentiality of the 5-HT2A receptor. And so Dr. Urosh Laban is a synthetic medicinal chemist with expertise in molecular biology, natural products, and genetics. A leading figure in psychedelic research, he earned his PhD under Professor David Nichols, synthesizing psychedelic tryptamines and dopamine agonists to explore treatments for Parkinson's, schizophrenia, addiction, and cognitive disorders. His research centers on serotonin dopamine agonists and their therapeutic potential in neurodegeneration, depression, dementia, epilepsy, and obesity. Dr. Laban specializes in drug discovery, hit-to-lead optimization, structure, and fragment-based drug design, and GPCR modeling, which we'll talk about today. He also works on scaling compounds for clinical use. In Brazil, where he currently lives, he helped develop APIs and synthetic processes for major firms like Laborvida and Europharma, contributing to treatments for Alzheimer's, PTSD, inflammation, and cancer, while generating multi-million-dollar value. So I'm really excited to have Dr. Laban with us. Hi, Dr. Laban, how are you doing?
Hello, Alex. Thank you for inviting me. Welcome, everybody.
Yes.
I'm very honored to be here today.
Sure. So why don't you, take us through the world of the 5-HT2A receptor? Mm-hmm. Why do you think it's still such an interesting receptor and the potential, therapeutic applications that it can lead to?
5-HT2A has become really popular last 5 years. We're undergoing psychedelic renaissance, and we're finally slowly figuring out what it does. It's an important receptor in the brain, in the body, in the lungs, in the stomach, but it is very antidepressive. It is neurogenic, anti-inflammatory. So we're just finding out these therapeutic benefits of these drugs, and we're trying to make them non-psychedelic now. So we can take a look a little bit how that all looks like. Let me just share my screen.
And it's interesting that with the study of the 5-HT2A comes this new concept of biased agonism.
Biased agonism, we'll talk about that a little bit today.
Yes, wonderful.
Okay.
All right, let's dive into it.
The slide. Okay. So years ago, before I even got into this field, I had a good friend who told me, so Urosh, when I take these compounds, it's like someone took off the spider webs off my brain. This was in the 90s. It's like running an antivirus and then restarting the computer, and I feel so much better, so much clearer. What the native people have known for centuries, we're just finding out now that Mother Nature provides us with amazing cures for depression and the maladies we suffer in the modern world, and we're just finding these things out now. To me, the most important aspect of these drugs is that they promote neurogenesis, synaptogenesis, they're neuroprotective, anti-inflammatory agents, and do many other things. But okay, let's take a look. Today, the main antidepressant, our gold standard, is escitalopram, and you have to take it for at least a month to feel the effects. And I work with a physician who routinely prescribes these compounds to cancer patients. And I talk to them also. They say, Urosh, I don't feel anything. I forget that I have cancer. Everything turns into this blur, right? And the recent studies show that psilocybin has the same effect, but it's immediate. It's robust and long-lasting, and it can be used for depression, for alcoholism, for tobacco addiction, for heroin, cocaine, whatever, right? So after a single dose, we see immediate, profound, and long-lasting effects in these drugs. Now, psychedelics are just one second. Psychedelics are some of the safest substances that we know of. There has never been recorded an overdose with LSD, mushrooms, or mescaline, right? But ketamine has problems. Ketamine is highly addictive, and this things taking ketamine nasal spray at home without medical supervision will not turn out right. We all know how John Lilly ended up. 5-HT2A is most densely concentrated in the brain, in the prefrontal cortex. We have it in the lungs, in the stomach, and PFC is the probably most interesting and important part of the brain because it governs and that is how we interact with our environment, with ourselves. It is the center of cognition, emotion, maybe, I don't know, consciousness, something like that. But it is the brain, it is the part of the brain that evolves, that is different from other primates. It is probably the most developed region of the brain. 5-HT2A, there are many serotonin receptors, right? I'll mention 5-HT1A because it always comes back to haunt me. That was my first project. I also am interested in 5-HT7A agonists because they are upregulated in the hippocampus after psilocybin administration, and we see neurogenesis. So that's what really interests me. And here we see the GPCR. So 5-HT2A is a 7 transmembrane protein, 7 helices that spans the membrane. It's a signaling protein. Upon ligand binding on the extracellular side, we get the conformational shift in the intracellular side, and then we can get various activations of various secondary messengers. Today we'll focus basically on G-alpha and Beta Arrestin, but there's also these visual effects that everybody tries to stay away from, and potent anti-inflammatory effects. Okay, and this signaling is called functional selectivity or biased agonism. And all the psychedelic drugs that produce hallucinations basically signal through this GQGI signaling, and they produce a head twitch, meaning that they're psychedelic. Beta arrestins don't do that. They're non-psychedelic, but they're slower acting, and we'll talk about that a little bit. Polymorphism. Well, how we react to antipsychotics, and especially psychedelics, is determined by maybe a single point of mutation. Some people will be more or less sensitive to psychedelics. Some will react better or not as good to fluoxetine. This just came out, this paper. I didn't put anything in, but it has to do a lot with genetics, with inflammation. There's about 19 genes that are differentially expressed. So folks should really take a look at this paper. It's beyond the scope of this presentation, but it's important. Okay, so these single point mutations, there are a couple on the extracellular side, a few of them on the cellular side. They can be gain of function, loss of function, or neutral, like isoleucine to valine won't have much effect, but this histidine 452 to tyrosine, if you go from histidine to tyrosine, you completely change the chemistry of the amino acid. The histidines are important in A-beta and synuclein because upon binding with copper, they produce these toxic intermediates. Tyrosine, on the other hand, can be phosphorylated and can do completely different chemistry. So just a single little change can influence completely the tertiary structure and the downstream signaling of this receptor. Okay,
especially since it's intracellular, yeah.
Here you can see the mutagenesis data. We'll talk more in detail about not all of them, but some of them that are more important. And okay, but before talking that, we need to figure out how to make a selective 5-HT2A agonist, right? This is all from crystal structures, and we can see that there's most of these tryptamines and psychedelics are not non-selective, right? And we especially want to stay away from 2B activation because it is involved in not only in cardiac valvulopathy. It's like the top three killer diseases. This 5-HT2B is involved: heart disease, cancer, and number three is lung fibrosis. So that I had no idea, but it's also involved in breast cancer, pancreatic cancer, diabetes, and many other things. And the FDA issued a statement that they will forward any drug to orphan drug status if it's devoid of 2B. So that's another big thing. Here we see serotonin binding to the active site, and
looking from above.
Yeah, okay. Aspartate and serine 242, you will see always. They're the anchors. There's a covalent bond there, and you will see the phenylalanine in 340 and 339. They're highly conserved receptors, and also worms even have them, and they kind of do this pi stacking, and they orient the receptor properly. And this asparagine 343 finally showed up. This wasn't seen in the early crystal structures, but I'll talk a little bit about that.
So here we have a structure, sorry to interrupt you, but here we have a structure of serotonin in its receptor, and there's no hallucinations, but it's very similar to the psilocybin, or a psilocin, should I say. So we'll try to understand which interaction might be responsible for potential hallucination.
Serotonin is an endogenous ligand. I mean, we have it, it was first found in serum. That's why it deserves the name, but it is not psychedelic.
And there's also the transporter that, the serotonin transporter that
we start talking about cert, that's a whole other story, because some of these compounds, especially if you put the methoxy group on psilocin, it becomes a cert inhibitor. And it has a little kind of antidepressant-like stimuli if you read the reports online. So I think that is relevant.
But the time of how long serotonin stays in its receptor is very brief compared to
because very small
psychedelics.
You don't have the methyls here, so it is metabolized very quickly. There is like an oxidative deamination, I think, that happens that leaves an aldehyde here. So turnover and metabolism of serotonin is very fast. Okay. Psilocin, the first crystal structure. Similar binding. Roth just published this, and there's another threonine here, which I really like. So there's another anchor, and there's a whole set of hydrogen bond interactions in the bottom of the receptor. I'll show some. If you dock LSD in D2, you will see like 6 or 7 all interacting together. So this is very, very valid. When I was in grad school, I thought the serine 159 participates, but it wasn't that. Back in the day, I would cut out psilocin, I would draw the thing, and I would manually try to look. But today we have computers, so it's much easier. Mentioning this is the Hurg value, right? Hurg is a potassium channel in the heart, and that causes cardiotoxicity, and that's the things we want to stay away from. We try to keep this number low. Glide shows the affinity for the receptor. So this is around 10. It is 5-methoxy DMT, psilocin, LSD is around 11. So we can get kind of an idea how potent the binding will be, and the experimental data translates nicely into my model. Okay, so we see now. In rodents do not have serine, they have an alanine, T242. So this hydrogen bond is prevented. So there is no interaction with serine 242. Glennon made a bunch of 7 substituted tryptamine, 7 bromofluoro, halo, and showed that in the rat receptor they have a higher affinity, but not in the human. So rats maybe trip differently, I don't know. But I caught this interaction and I found it interesting again, asparagine 343. So when we always test these compounds, we always have a human line and a mouse line, and sometimes we see deviations. Okay. Now, how to probe the receptor? How can we learn more things about certain amino acids? Well, we can design ligands that specifically interact with a certain residue. And In this case, I developed this 4-substituted tryptamine, and it's basically molecular tweezers that interacts with this asparagine. And you can see the mutagenesis data depending on the ligand, what it does. But take-home message, this is a crucial residue involved both in GQ and beta arrestin interactions. And these compounds should be maybe 10 to 50 times more potent than LSD. If you want to make them non-psychedelic, just take this nitrogen alpha. We'll talk about it in a second.
Mm-hmm.
And what I keep seeing over and over is
okay.
flips of compounds. These azaindoles and pyrazoles were a set of about 30 compounds, a company that wanted to sell the IP. When I modeled this, I saw that there's no brain penetration. Log P is about 1. It needs to be 1.8. You could make prodrugs, and yeah, they would be fine. But the most interesting thing out of all the compounds, only these two, 7-azaindole and the pyrazole, this would be even more antioxidant than the tryptamine, they kind of invert and flip. And instead of serine 242 binding N1, it starts binding 4-hydroxy. And it's only 2 of these compounds. Okay? So I did make and I've noticed this with norpsilocin, monoisopropyl psilocin, things like those. So we made this compound, we crystallize it, and we found out that in the crystal structure, there's this ergoline-like conformation, there's an intramolecular hydrogen bond. And this probably happens in psilocybin. This interaction prevents oxidation and gives longer half-life to the compound. And here you can see, resolution is not the best, but you can see aspartate stabilized by this serine 159 and serine 242. Also, there is a extended hydrogen bond interaction with glycine. So I found this interesting. Can we watch the video now?
Yes.
Part of the program in studying the monoalkyl tryptamines. And here we can see the crystal structure. Can you scroll down a little bit just to show the synthesis?
Yeah.
Okay. Yeah, so we started with 4-benzyloxindole, did this Peter Anthony, reduced it with diborane, and then debenzylated, and we obtained the crystal structures of all the intermediates, and we should look at that today. So the crystal structure of this compound is basically 4-hydroxy in the 4 position with forming a intramolecular hydrogen bond with a nitrogen. And this is the so-called ergoline conformation, and this is the non-ergoline conformation of psilocin analogs. So the ergoline conformation resembles ergoline's LSD, and this one has the side chain pointed away from the 4-hydroxy. These compounds tend to be a little bit more selective, and the compounds that have the side chain in the ergoline conformation tend to bind serotonin 5-HT2-1A and dopamine D2 receptors. Most interesting thing is that we do find a conformation similar to this one in the receptor. Actually, this is not the main binding pose of psilocin, but we can see that also an ergoline-like conformation with an intramolecular hydrogen bond, all stabilized by another hydrogen bond from this asparagine 343. And if we can superimpose it, we can see it's very similar binding pose, except the side chain is in the alpha versus the beta position. My other video explained how psilocin binds most commonly. We can just rotate this dimethylamino 180 degrees, and then it will assume a bond with aspartate 155. In addition, there is a hydrogen bond to serine 242 with most tryptamines and LSD utilize this linkage. The most interesting thing is the tryptamines are symmetric. So you can kind of superimpose them and put nitrogen on the oxygen, oxygen, nitrogen, and you achieve C2 symmetry, and these side chains can bind in approximately the same position. And what we've noticed when we do modeling, that these monoalkyl tryptamines, norpsilocin, N-ethyl, propyl, isopropyl, and butyl, they do not bind in this conformation, they flip. Okay, and that's what I find really interesting. So the 4-hydroxy binds to serine 242, and then the side chain binds to aspartate. The binding energies are about the same, But what implication this has on the pharmacology is still yet to be seen. This might give rise to functional selectivity, agonist trafficking. Maybe in the presynaptic, there is a different binding mode than in the postsynaptic sites. But this is also possible, right? So we won't know until someone grabs this and crystallizes it. But till then, it's only a hypothesis. The anisopropyl psilocin, we crystallized it as a free base. The hydrochloride salt is unstable. The fumarate is completely insoluble in water. It has solubility of 1 mg per mil in DMSO. But basically, these are novel classes of tryptamines that we're examining, and I thought that this would be interesting to show to our audience. Thank you.
Fantastic. Fantastic.
Good. Let's continue. Okay. Okay, here we come to the king of psychedelics, LSD. Okay. Okay. LSD is only a partial agonist at 2A, and it's one of the most potent and long-lasting psychedelics. It has very slow binding kinetics. It is metabolized to 13-hydroxy-LSD, which is dopaminergic, and that is why some folks may have bad trips. There, I have another video of that. And it has very complex polypharmacology. It hits 20, 30, 40 receptors, adrenaline, serotonin, dopamine, a bunch of other things. The interesting thing is we see the binding pose, again, the same thing, phenylalanine, serine, aspartate. Up here, there is leucine 229, 228. There's another phenylalanine stabilizing the whole thing. Once LSD binds, this leucine 229 acts like a cap. It shuts on top of the receptor, and LSD stays for 6-8 hours. That's why it's so long acting. So the kinetics are really, really slow. And I played around when the crystal structure first came out. I took Brian structure 6WGT. I modeled it, minimized it. You can see cholesterol, lipoic acids, water. You have to clean it up completely. And then I manually picked a bunch of amino acids. They're all covalent linkers that I see that they come up over and over and over again. So I took one of my best linkers, docked LSD, and I caught a couple of new interactions here, notably the leucine, carbonyl. This is maybe less than a kcal per mole. It's not a very important interaction, but if you imagine LSD locked into the receptor, the only rotation that can occur is this side chain, then this becomes very significant in the receptor active site. And I get some additional bonds here. I pick up asparagine, leucine, phenylalanine. So if this is viable, if this really happens in the receptor, has to be seen, but I get good document results here. Okay.
And you think the lead is the interaction with the lead could explain the 6 hours retaining?
The lid closes and just traps the LSD inside. I have another video that we can show if we have time in the end, but there is a lid on top, LSD binds, and then the whole thing closes. And it's like a lobster trap, something like that. Once it comes in, it doesn't come out for a long time. And yeah, we have the semi-open structure now, but the semi-open is, I think, more important for small molecules that have a high on-off rate, like DMT. And we still don't know. We have to look at those things more carefully.
Does that happen at other GPCRs
I don't know. I know that LSD does that, and it does it really well, but I'm not familiar with other GPCR, and GPCR is what LSD does. Probably there are compounds like that, definitely. GPCRs have homology. So now we can design ligands that can probe certain amino acids, and if one makes this LSD analog, and be more potent, and we can probe this serine 131, and serine 131 is very important, conserved all across the receptors, and it forms part of this ligand recognition triad, right? Aspartate, the toggle shift, and the other things. If you mutate this, there is a reduction of serotonin binding for about 18-fold. This is the only mutagenesis data that I found. You can make another one, morpholine, probe this 363, and I see this residue coming up over and over again.
Yeah.
Again, more potent, and we don't know, there's no mutagenesis, but again, this is very important. This is way down in the TM7. This has to do with mini GQ and maybe arrestins. It doesn't directly participate, but it is there. There's a German group that synthesized a bunch of these things. Some of the compounds are similar that I show here. Okay, Morphled, yeah, take the red pill. Key bias switching mutations, serine, asparagine, the toggle shift seems to be the most important. That gives this beta arrestin bias, and we'll talk about that in a second. Serine 159 is important. You will see this in these beta arrestin ligands. So it has a GQ bias, and this just came out a few years ago. And the M-bombs. Okay, the M-bombs are just benzylated phenethylamines. And this was made in Nichols group, but the synthesis of this compound, the DMB, is very difficult. It has 4 diastereomers, and only one is really active, this SS trans compound. But when you do pharmacology, you will see that this compound has no 2B2C. So I immediately docked it, placed it over LSD, and what I see, this is about 3 years ago. There is no serine 242. So if you avoid the serine 242, you can get 5-HT2B selectivity and remove hallucinations. So that was very interesting. Now with AI, you can figure this out in a day, what it took me a year to figure out. And here's the binding. I get few other residues in there, but you can see the aspartate 155 and the 159. Okay, so they kind of freeze this N-hydroxybenzyl in a certain conformation, which I'll show in a second, right? So you can see the toggle switch, you can see the tryptophan. And what is the mechanism of these beta arrestin ligands? They kind of inhibit the movement of the tryptophan here, so inhibiting GQ signaling. For many GQ to bind, this tryptophan needs to be completely open for the GQ to bind, but for beta arrestin, that is not necessary. So beta arrestins kind of trick GQ. They freeze this conformation and they favor intermediary transition states that kind of inhibit the GQ signaling. We'll see that in a second. Okay, this is the list of compounds with beta arrestin bias. The M-bond, the TBG, the phenethylamines, and then we have the GQ bias. But they're not very selective. They're maybe biased by 20%. There's still considerable beta arrestin signaling. And okay, we can see DOI here. Brian's compound, the Z7757. This pyrimidine motif is present in 2-3 compounds that I find that are neurogenic hippocampal activators. So they produce neurogenesis in the hippocampus without hallucinations. The biggest mystery to me was 2CB, TCB2, which supposedly is non-hallucinogenic, but it is a full and selective 5-HT2A agonist. It causes some hypothermia. And when you see hypothermia, immediately I think of 5-HT1A. So what could be going on? Well, I think that this can be a 5-HT1A allosteric modulator, maybe activating 1A a little bit and dampening the hallucinations, because 1A inhibits the action of 5-HT2A receptor. It also alters this receptor heterodimerization, because 2A can dimerize with 1A, with D2, with a bunch of other receptors and perform some kind of signaling. I don't even know what it does. But the het twitch is about half of DOI or LSD. So this is my only explanation to why it is not hallucinogenic. Another compound, this is Brian's compound, GQ selective. And for GQ, you don't see much difference here. You see serine, you see threonine, the same as in psilocin and the side chain. With these compounds, they do something very special that you cannot see in the active site. They promote conformational changes favoring one or the other, right? So in the active site, there is very little to see. And I kind of played around with these compounds at Purdue. I made an analog of RU24969. I still think that these compounds, these THPs, may be a little bit dopaminergic in 1A, and no one has tested them yet. When I tried to put the nucleus of the most potent and selective 5-HT1A agonist, which is 4-fluoro-5-methoxy. The whole thing fell apart, even just to potassium carbonate. It underwent some retrodielsaldr and just decomposed. And fluorine really complicates synthesis sometimes. And here we have the beta arrestin ligands. It started with the MBOH, and then you start making alterations on the side chain. And the most interesting thing, the first beta arrestin favoring tryptamine is the DALT. And immediately you notice the same thing, acetylene, olefins, their structural features that are similar. I need to model this and look at this. DALT, if you try to reduce this with hydrogen palladium, it won't reduce it. The allyl group will just fall off. So it's like, and in the industry, we like to avoid allylamines because of their toxicity. But as a research chemical, this is very interesting. And here you can see structure activity, how they made these arrestins. These are McCorvey's compounds, the nitro. Nitro is also toxic. It will never be like a nice drug, but it's a very interesting research chemical. And the RS compound favors beta arrestins in a very unique way because it sends this tryptophan in a completely new conformations that we haven't
downward
seen. Yeah. And this is the beta arrestin right here, right? And it does that. It plays around with the toggle switch, but the most interesting thing that it kind of not destabilizes it. It forms like a wrong transition state for the GQ to bind. So it behaves maybe like a beta arrestin antagonist or something like that. So this is the pharmacology of these new compounds. And here we can see the crystal structure in the glide model. Again, we only see aspartate and the phenylalanines. All the rest of the hydrogen bonds are lost. And so finally, if we can summarize this, what is the difference between GQ and beta arrestin? All the GQ selective ligands are very potent and selective, 2A agonists. Beta arrestins are less potent, they're like in the micromolar range. They both bind in the orthosteric binding site. And as I said, the GQ ones have more hydrogen bonds, very well defined, and these bonds are lacking in beta arrestin. Pi stanchic preserved, yes. Rotation of the toggle, yes. For GQ to bind this amino acid needs to move about 14 angstroms. It needs to be completely open, while beta arrestins keep the toggle switch occupied and kind of shut. Semi-open receptor, yes, I think that classical psychedelics, at least the small ones, can bind, but beta arrestin, no. And there is a time scale that Brian just published, that there is a delayed effect, the time release of beta arrestin. They start pumping the signal after like 6 hours. And how to solve this problem by molecular modeling? If you look at the inactive protein, this would be like a global minimum. You will find if you rotate the dihedral angle, you will see maybe a couple of global minimums. But you can see in the GQ bound state, this needs to swing open about 60 degrees, full outward forward tilt that will produce G protein. Coupling, psychedelics, head twitch. In the beta arrestin bias, the toggle switch, there's very little movement, some maybe on the wrong side, there's a partial tilt. And the effect of that is that we get beta arrestin recruitment and no GQ. So in order to kind of model these compounds, we'll have to freeze this toggle switch, then do modeling, Monte Carlo, put in ligands, or do it without ligand free, then add different ligands. There is a way to do it. I'm kind of getting ideas how we can make a predictive model, just looking at the GQ and beta arrestin models. And to finish, there are two schools of thought, non-psychedelic versus psychedelic. I cannot say that I favor one or the other. Both of them are important, but with classical psychedelics and these new things that we have made, you can't go wrong. I mean, we have shown that these things work very fast, very quickly, and immediately with these non-psychedelic psychoplastogens that also makes. I think you will need a couple of weeks for the drug to kick in. They will act more like these piracetams, the nootropic drugs. The BMB 201 that we made is a partial agonist, so we reduce the hallucinations. We can see psilocybin full agonist and long-acting and hallucinogenic, and the BMB-202, which is a psychedelic that lasts under 2 hours, that we managed to make a very quick, short-acting drug that can be done 2 hours in the doctor's office. And to summarize, the most important slide to me, I don't care if drugs are psychedelic, non-psychedelic, if they regrow your neurons, that's all that counts. So if you look at brain-derived neurotrophic factor, ketamine, DOI, they all produce neurogenesis, that are partial agonists also, but if you look at BMB-202, it's almost equipotent to BNDF, and that is very, very, yeah. And this just came out, the intensive psychedelic experience is reliably associated with clinical improvements. So we know that classical psychedelic works, high doses. If you ever seen like a patient who is with MD, with major depression, or folks who come back from the war, and you start giving them regular antidepressants, the next day the guy may jump off a bridge, kill himself, do something like that. So I'm a proponent, if you have serious cases of depression, hit them with one of these psychedelics and then continue non-psychedelics or classical antidepressants. I mean, that's the way to go. And just to rectify myself from last time, for Brian and Sam, if they're here, the Gilgamesh compound, yes, 5-fluoro-methyl-ethyl DMT. And I made these compounds back at Purdue, and they're all potent 1A agonists. So what I think what is going on, we're producing a little bit of serotonin syndrome, which is dampening the hallucinations, right? Why did they put methyl-ethyl? Well, just to break the patent. And if you compare LSD, psilocybin, and the CZ74 is the diethyl compound. It has been examined by a German group back in the 80s, but they stopped because of bad trips of some of the subjects. These things, I think that they can be oxidized and this ester amide removed faster. And you see that in MIPT, DIPT, they're all shorter acting drugs, okay? So, and just to confirm this, I found out the data back at Purdue. This is drug discrimination. This is ancient Hetwich. And you can see wherever you put fluoro, there is no substitution, except for 6-fluoro-5-methoxy, which had some activity, we made the 6-fluoro-psilocin, that was our lead compound. That was interesting, but we gave it up. It had some pharmacology issues. So this is LSD. The same thing is with DOI. You introduce the fluoro, hallucinations drop. It doesn't substitute for LSD anymore. Okay, thank you. Questions? First year Nichols group.
When was that?
Oh, late 90s. Many good things came out of this hood right here.
Is that your magic wand?
No, this is Mickey Mouse from Fantasia. The big wizard is away.
We have a comment from Sam Bannister. He says, take a look. Yes, he says, take a look at IHCH 8134, currently in clinical development. I don't know if you heard of that compound.
IHCH. That's the one. I know. I have it here. Hold on. That is the Chinese group compound. Where is it? This one.
Oh, right? No. His is 8134.
8134. Man.
This one is 7.
Hold on. I'll put it on Google. What is the name?
IHCH.
IHCH.
Hyphen 8134.
34. Yeah. Okay. I know this compound. Yeah,
this one is 7086. I don't know.
It was built on an antipsychotic. It's non-hallucinogenic. I don't know if it's GQ or Beta Arrestin. Right? I have to take a look. But yeah, I looked at the crystal structure. I couldn't find anything. Again, if it's Beta Arrestin, you don't know what's going on. Right? There's not so much binding, but this also showed a very profound antidepressant effect in rats. Right, so this kind of resembles LSD, and then they put the linker up here, and you can see the where are those morpholads? The same thing as here, right? You kind of imitate the LSD, and you can keep building, and then it starts hitting this 363, 131. So you can put substituents here, but the bigger molecule it is, and the more hydrophobic interactions, you will favor antagonists, right? So for agonists, you need a lot of hydrogen bonds. For antagonists, they favor these greasy interactions.
And so, what would be the take-home message to differentiate GQ versus beta arrestin? Do you think it's the toggle switch, or?
Yeah. So basically, from all that I've learned, and I've looked at many structures, this toggle switch, that's where the bunny rabbit lies, in that bush. And if we could really somehow examine these interactions and basically do modeling, I would freeze this angle, then relax the receptor, then I would put ligands, and then I would dock and see if there's any shift, anything that goes on. But just by freezing this toggle shift, you might get to see some differences between GQ and beta arrestin. You would get some favoring, but you would have to model the whole receptor, then look at the downstream, the helix 7, helix 8, how do they rearrange when these ligands are added. I haven't seen any experiments. I would do an experiment. I would add a GQ together with beta arrestin and see what happens. I have no idea, but things like those.
Wonderful. Well, thank you so much.
Thank you,
Dr.
Alex.
Laban, for your time. We don't have any more questions, but we should do another talk soon. So
yes. Let me stop sharing here. Hold on.
Yes. Thank you so much for your time.
Thank you, everybody.
And everybody, I hope you enjoy the talk, and don't hesitate to reach out if you have any more questions for Urosh Laban.
Thank you.
Thank you.
Bye.
Bye.